All right, thank you everyone. Today we're going to be presenting some of our work on weathering deep space and optimizing 5-AMP and 6N-cyclohexyladenosine to induce a state of controlled hypometabolism. My name is Dr. Eric Bredahl, and presenting with me today is my head undergraduate researcher, Nick Johnson. As you guys may have seen in reading through our abstract, our goal is to try to better understand and come up with countermeasures that can be used to overcome the microgravity-induced atrophy. Atrophy or tissue loss is common in spaceflight, and it's something that is usually, if not always, enzymatically based. So anytime you can reduce the energy environment or reduce ATP production, you essentially have the potential to slow metabolic activity and some of those enzymatic processes and delay or at least attenuate a degree of tissue loss, namely bone, muscle, cardiac, etc. To do this, we cultured a wide variety of cells that represent some of the tissues that are most affected by microgravity, and we worked by treating them with a variety of different doses of CHA as well as 5-adenosine monophosphate. Our results are intriguing. We found some really unique findings when it came to the ability of both 5-AMP and CHA to reduce metabolic activity.
As Dr. Bredahl mentioned, minimizing tissue loss during prolonged microgravity is key for long-term survival and preservation of functional capacity. And through the literature, we found 5-adenosine monophosphate and 6N-cyclohexyladenosine as both to be good adenosine receptor agonists that act on different subunits. And previous research in laboratory animals have shown that these can induce hypometabolic states. However, the dosing is quite variable, so our goal is to really identify the optimal doses that can create a hypometabolic state while still preserving cellular viability. These compounds, if effective, would really lower the required human inputs and outputs and free up capacity on the spaceship for extra fuel, research equipment, or just really reduce the overall payload. And the hypometabolic state would slow the rate of tissue loss while maintaining functional capacity. The cells we selected, those being muscle, skeletal muscle, cardiac muscle, kidney, neural, and bone, are some of the tissues that are most impacted by microgravity. And ultimately, these compounds represent a potential treatment avenue that has profound implications for human spaceflight.
To do this work, we cultured L6 skeletal muscle myoblasts, HFOB osteoblasts, and HEK293 kidney cells. We cultured these cells in a 90% DMEM, 10% fetal bovine serum supplemented with penicillin. We incubated these cells in a 37-degree cell incubator. They were in the cell incubator until they reached a confluency of 90 to 95%. During this growing period, we replaced the growth media every 2 to 3 days, and we continued this process until the cells reached a density of 10,000 or reached a density of 90 to 95% confluency, at which point we then seeded the cells onto a 96-well plate at a density of 10,000 cells per mil. We then allowed the cells to recover from plating for 24 hours, at which point we then treated them with a variety of different doses of both CHA and 5-AMP. The range for CHA was between 50 millimolar and 100 micromolar. For 5-AMP, we had a working range of 1 molar to 30 millimolars. The incubation period lasted for 12 hours. We assessed cell viability via a Nikon live silicon focal imaging system. We assessed glycolytic and oxygen consumption via ELISA-based assays. All experiments that you'll see today were run in triplicate.
So to start off, we'll go with these L6 results. For 5-AMP, we found that at 125 millimolar dosage, there was a high level of viability, very comparable to the control cells, and still having about 50% reduction in glycolytic activity. However, oxygen consumption remained unchanged throughout this. For the CHA, we have very little data at this point, and we're in the process of collecting more data. But with the data we do have, we found that at 12.5 millimolar, there was a moderate decrease in glycolytic activity. And just a visual representation of the viability. The red cells here in these pictures are considered dead, and the green ones are viable. And as we go from left to right, we go from high concentration on the far left of 1,000 millimolar to one of our lower concentrations of 30 millimolar on the right. And you can see the large increase of green cells throughout these pictures, and that's shown in our graphs as well.
Continuing on with our HEK293 cells, and these are representative of our kidney cells, what we found was kind of interesting. We saw that a dose of 500 millimolars of AMP, 5-AMP, provided the greatest effect without negatively affecting cellular viability. At this dose, we reduced oxygen consumption by almost a fourth, and we saw more than a 60% reduction in glycolytic activity reduction. There was no significant change at viability. Similarly, at a dose of 50 millimolars, we saw a 10% reduction in oxygen consumption, whereas we saw a slightly lower reduction in glycolytic activity than that of 5-AMP, but still noteworthy. So again, at a dose of 50 millimolars, we were able to suppress both glycolytic and oxygen consumption without negatively impacting cellular viability.
Then for our HFOB cells, these are our bone cells. Like the kidney cells, the 5-AMP optimal dose was 500 millimolar. Viability remained pretty steady at the control levels throughout all the doses, but at this 500 millimolar, there was about a 20% reduction in oxygen consumption. For the CHA, just like the L6 muscle cells, the optimal dose we found here was 12.5 millimolar. Viability once again remained fairly high throughout all the different doses, but at this 12.5 dose, there was a 15% reduction in oxygen consumption. We are still doing data collection on glycolytic activity, but in the next few weeks, we should have that.
Ultimately, what we found is the rate-limiting tissue that has the most sensitivity to both CHA and 5-AMP is that of the L6 myoblasts. Its optimal dose was 125. So as such, we're going to consider that our rate-limiting tissue, because at a dose higher than that, we start to lose muscle mass, which is not a good thing for astronauts traveling through space. However, at a dose of 125 millimolars, we were still able to produce measurable metabolic suppression in all of the cell lines studied. Similarly, a dose of 12.5 millimolars of CHA will also produce a moderate degree of metabolic depression without negatively affecting cellular viability. Moving forward, we plan to continue our analysis of different cell lines for blood of neural tissue, testee tissue, as well as more cardiac myoblast tissue. Hopefully, we'll be able to better understand how these treatments affect those cell lines and maybe be able to refine our optimal dosing for both 5-AMP and CHA. Furthermore, we want to extrapolate this work into a laboratory animal to better understand how these compounds affect metabolic consumption at the macro level. This work was funded by a NASA EPSCoR grant, and I'd also like to take a few moments to thank my research team, especially my undergraduate research team, the NASA EPSCoR office, the Creighton Biomedical Imaging Corps, as well as the Creighton Medical School. If you have any questions, please feel free to contact me at ericbredahl@creighton.edu. You can also phone me at 402-280-3471. Once again, I'd like to thank you all for taking some time out of your day to listen to this presentation. Have a great day.